Hello and good day to everyone. I am Teng from UNIMAP. The title for my research is about liquid pineapple waste for biobutanol production. Nowadays, the fuel is getting expensive. Air pollution and yet the environmental issues also getting serious. Is there any ways to produce biofuel from renewable sources? Like producing from food wastes like pineapple peel. It is good to produce biofuel from renewable sources as it can help to reduce environmental issue caused by the excessive use of fossil fuels. And now let's further discuss about that.
Production of biobutanol as biofuel is rising due to high cost of fossil fuels and environmental issue like air pollution. Liquid pineapple waste, LPW, was chosen as raw material or feedstocks for biobutanol production in this research. LPW will cause serious environmental issues if they are discharged to the environment without treatment, while the cost to treat LPW is expensive. In this project, the sugars composition in LPW were analyzed and the significant factors on biobutanol production were screened with the use of placket Berman design, PBD. It was expected that LPW from food canning industries will be one of the promising carbon source for future biobutanol production in Malaysia.
There are total 17,165 metric tons of canned pineapple was produced in Malaysia in the year of 2011. There is about 30% to 50% of the total pineapple weight are dumped as waste as they are the unused part of pineapple. The unused part of pineapple include of the stem, crown, core and peel. They are removed and considered as waste during pineapple processing. Liquid pineapple waste contain high concentrations of sugars such as sucrose, glucose, fructose and other valuable nutrients which help in enhancing biobutanol production. Therefore, the liquid pineapple waste have the possibility to be recycled and reused as raw material for other industries, or converted into useful and profitable products like biobutanol. Next, is the project description. In this project, the characterization of liquid pineapple waste, especially the specific sugar composition which include of glucose, sucrose and fructose, are determined by high-performance liquid chromatography, HPLC. This table showing is the concentration of glucose, fructose and sucrose in LPW determined by using HPLC. The concentration of fructose was the highest in LPW, which contained 33.81 plus or minus 0.43 gram per liter. Followed by glucose, which contained 19.64 plus or minus 0.43 gram per liter, and sucrose, which contained 9.53 plus or minus 0.36 gram per liter. Meanwhile, the total sugar concentration in LPW was 62.99 plus or minus 2.15 gram per liter.
The concentration of sucrose, glucose and fructose might be different due to the different types and sources of pineapple used include the different of pineapple canning factory. It is also because of the species, soil condition, and the maturity stages of pineapple fruit. The sugar contents of pineapple were changed in different harvesting seasons as sucrose phosphate synthase activity was also changed in different seasons.
Next, the significant factors on biobutanol production were analyzed with the use of placket Berman design, PBD. PBD was used to screen few important parameters that will affect the production of biobutanol. There were total 11 medium nutrients being analyzed. The graph showing is the Pareto plot which reflect the effects of variables on biobutanol production. The red color bar are the factors having positive effect on biobutanol production, while the bar in black color are the factors having negative effects on biobutanol production. Based on the Pareto plot, the greatest to least negative effect on biobutanol production were ammonium nitrate, ammonium acetate, yeast extract, KH2PO4, tryptone, thiamine and MgSO47H2O accordingly. The factors with greatest to least positive effect were cysteine, MnSO47H2O, FeSO47H2O and K2HPO4. To increase the biobutanol production, the concentration of the negative effect factors should be decreased, and the concentration of positive effect factors should be increased.
Based on the result, ammonium nitrate, ammonium acetate and yeast extract were the most significant factor on the production of biobutanol. Ammonium nitrate and ammonium acetate were the inorganic nitrogen source that showed high negative effect on biobutanol production. These ammonium salts were added into the fermentation medium and act as promoters for solvent like biobutanol production, and also as buffering agent. Yeast extract, which is rich of minerals, amino acids, vitamins, growth factor and proteins, was used as nitrogen source for fermentation and cell culture. Yeast extract is important to accelerate biobutanol production due to its strong effects on utilization of sugar during biobutanol fermentation. For novelty and inventiveness, liquid pineapple waste is a low-cost raw material as it is direct getting from the canning industry waste. This is an aerobic process for biobutanol production as the microbes used can be survived even under aerobic condition. It is also a simple process of biofuel production without any pre-treatment process. It does not need any special bioreactor and setup.
Lastly, for commercialization, biobutanol is a renewable energy as it is converted from biomass, which is a renewable sources. Biobutanol has better fuel properties as compared to other biofuels. Biobutanol is more environmental friendly and it may help to reduce environmental issue caused by the excessive use of petroleums. Whereas, liquid pineapple waste is a cheap carbon source as it is a waste from pineapple canning industries. Thank you for watching and have a nice day.